Well, my clock says noon, so it is my pleasure to welcome all of you to Responsible Reopening, Best Practices for Testing and Contact Tracing, which is being co-hosted by the Quinlan School of Business and the Parkinson School of Health Sciences and Public Health. I'm going to give you a quick introduction to what we'll be up to today, and then I'm going to turn it over to Dean Elaine Murado, who will be beginning this journey of how you can be part of responsible reopening. Let me just start on behalf of the Dean of the Quinlan School of Business, where I sit, to say how grateful we are for your joining us. As we looked over the list, we saw people that are truly on the front lines of figuring out how to reopen in a way that is safe and successful. We saw people at nonprofits who are doing so with their clients in mind, and people at businesses who are making sure that their businesses can continue to sustain and can employ and can help to create vibrancy in our economy in this moment. The origin of this series that this webinar is a part of is part of a broader question that Loyola as a whole and that our schools in particular we're looking at in this moment, which is how can we harness our assets in this moment to be part of the solution? And obviously, step one was making sure that we were there for our students and helping them through this moment. But step two was how do we leverage our assets and begin to truly share with our community partners in business and in health and in the nonprofit community to ensure that we are giving all that we can in the asset that we have, which is knowledge, to help really ensure a safe and successful relief and now reopening. And so thank you for joining us. We look forward to having many of you back who were here for the first webinar on cross-sectoral partnerships. And we hope many of you will join us again in a week when we discover how to look at this from a human resources and operations perspective. But for now, we're going to look into testing and contact tracing, and I'm going to turn it over to the founding dean of the Parkinson School of Health Sciences and Public Health, Elaine Murado. Elaine?
I'm going to switch screens real quick. Okay, all right. Should be seeing it well. So thank you very much, Seth. We're delighted to be here again, partnering with you. And really it's a journey together. It's something we envision of how do we make sure we're bringing the best knowledge to the moment. And right now we're at a moment in the Chicagoland area of reopening safely and what's needed or necessary to do that as it relates to testing. I'm going to just share a couple of slides I shared in our first opening. And one is to remind us what motivated or excited us to be coming together and working with a public health school and a business school. And that's largely grounded in the fact, as we all know, that our health system here in the US is really a mosaic of interdependent actors and agencies, which encompasses employers, schools, families, individuals, our public health systems, our healthcare systems, our government agencies. And if we are to work together to improve our population health, it requires us to come together. And no time more than ever in the present than our response to COVID-19 requires us to think differently on how we might partner with one another. Last month, I shared with you a roadmap to recovery that was developed by the American Enterprise Institute. And they itemized some phases in very broad strokes, from slowing the spread, flattening the curve, to reopening safely, all the way to rebuilding our readiness. And this roadmap is going to provide us some general guidance. But in the last month, we've been given some more specifics. And hopefully you saw when you registered a link to Governor Pritzker's Restore Illinois plan. This is how that general roadmap is being adapted and reframed for our own Illinois experience. Again, thinking of it as phases. The first phase in terms of what is flattening the curve and really our strict stay-at-home policies, all the way to various phases that allow us to open up slowly, have larger gatherings, and get back to where things had been eventually. Where are we right now? If you're following the news, Illinois is somewhere in between phase 2, not quite phase 3. Most of the state is in a position of being able to move to phase 3, whereas Chicago itself is more in phase 2. So let me share a bit more as to what does that mean? What is the public health approach that allows us to move from phase to phase and to safely reopen our state? And this comes from the governor's plan. And you can look at it as maybe 3 streams. One is looking at what are we observing as our public health data regarding our cases, our healthcare capacity, as well as now advancing our capacity around testing, tracing, and isolating. And for us to continue to reopen safely in the Chicago, the markers that are being looked at by our mayor, Mayor Lightfoot, is really looking at these individual pieces. So I pulled together for you some highlights, one from her speech that she gave to the Economic Club of Chicago last week, and looking at the data this morning. Where do we stand on those elements? And I think this is important to remind ourselves. Reopening is not like turning off a light switch off and on. It's turning up the brightness, thinking of it as a dimmer. And as she and our public health leaders in the Chicagoland are looking at the data, we're seeing positive movement on metrics such as hospital capacity. But we have a long way to go in terms of our testing capacity. And that's the focus of our 2 guest speakers today, is to make sure you all have that knowledge 101 of what do we mean when we say we need more capacity for this. And really, by having a stable public health testing, that allows us to have a stable and resilient economic environment too. So it's in all of our best interest to be working together. So I pulled together some statistics from this morning. I find if you aren't already looking, the New York Times provides some excellent overview if you'd like to see where the rates are. They're pulling from other data sources like Hopkins, but put it together in visual ways that help us look at our own curve. And it's nice because it allows us to put it into comparison. If you look at Illinois, you might say we are sustaining a case rate. And haven't quite turned the corner in terms of it going down if you're looking at sort of our 7-day average. Within the Chicagoland region, there is still a need to be boosting our testing capacity by almost 50%. That's equivalent to about ensuring we have the capacity to test about 5% of residents per month. Maintaining what we'll call a low virus positivity rate. So of those being tested, how many are showing up positive and having disease? We're just now getting past that threshold of 20% that the state has set. The good news is that we're not seeing an increase, and so it's actually a slight decline. And continue to see our decrease in hospitalization. These rates are looking better, but just to put some context here, if you look at ICU and medical surgical beds capacity in the rest of the state, they have more of a 30–40% vacancy rate. So still with a focus in our own Chicago area to make sure we have that surge capacity. The good news is that there appears to be a good supply of ventilators and such. But what we wanted to focus the rest of our time with you is around the idea of an expanded system and testing that will allow us to have the foundation to do more community investigation and contact tracing. So Public Health 101 and infectious disease control in outbreaks is to bring the cases down as low as possible so that when you're looking at outbreaks with testing, it allows you now to understand who else has been exposed to someone positive and how you might isolate them as a method of control. And so that brings us to the topic today and our 2 guest speakers, who you'll be delighted in listening to their experience and expertise. Today, we're focusing what I might call as our toolbox for making this happen. How do we get Illinois into this phase 3 of recovery, in ensuring we have vibrant systems for surveillance and this contact tracing and isolation. We're talking today about testing 101. Next week is, as Seth mentioned, is the HR systems, policies, and systems operations to make sure it all goes. So think of it as a part 1 and 2. Today we're talking about what are our tools in our toolbox that we need. And we have 2 speakers with us today. Dr. Cameron Mirza, who will be giving us a briefing on Laboratory Testing 101. So it's a chance for you to ask any question around, so how do you know what's a good one? How do you know what to believe? He directs our medical laboratory science master's program in our school and is also a professor in pathology and laboratory medicine at our Stritch School of Medicine. He'll be talking first and then joined by Jill Taylor. We're delighted that she's making time to join us. She's the director of the Wadsworth Center from the New York State Department of Public Health. New York has been in the news and on the forefront, and I think there's much for us to be learning in Chicagoland in terms of public health's role in a data-driven reopening. She has worked extensively, not only in the state of New York, but also at a federal level. And I just list here, her involvement at the CDC Board of Scientific Counselors in the Office of Infectious Disease. So it's an opportunity to learn from others outside of our area and what we might bring into shaping our own resilient response. So I'll end there and turn it over to Dr. Mirza to continue. Let's see, I think you can just share directly.
Can you see my screen, guys?
Yes.
Excellent. Thank you, Dean Morado. And thank you to the Quinlan School of Business and the Parkinson School of Public Health and Health Sciences for this opportunity. It's an honor and a privilege really to talk to all of you about testing and really to shed a light. And as Dean Morado said, I'll take you into the toolbox today. So in many ways, when we're thinking about testing and contact tracing, right, we come back to the word testing, right? Because even for contact tracing, there will have to be testing. And so what I thought would be really interesting would be to bring you into the laboratory with me for 1 second and kind of talk about the circle of trust that we kind of depend upon when it comes to testing in the laboratory. And I think that it's really important to think of lab testing as the pillars that are really holding up the entire public health kind of response to COVID-19. Because, every day, all of us, whichever sphere we're in, we're hearing about cases that tested positive, then tested negative. And, you're hearing things about this test isn't good or this test isn't good. And I think that it's important for everyone to understand that every test has its, limitations and its strengths. And I think that understanding the concept of testing, how we implement it, what happens in a lab, what type of virus it is, etc. While not going into the weeds too much, I think it might be helpful when it comes to formulating kind of the narrative for a responsible reopening. And so I think that perhaps the easiest way to explain this to you is to talk about that circle of trust when what you have and your physician has when it comes to laboratory testing. So bear with me while I take you to this circle a little bit. So a patient comes to their physician and says, XYZ are my symptoms, and it seems reasonable to perform a test. So the first step that you want to do when that test is actually going to happen is to decide what type of test is going to be ordered. Is that test going to be in the blood? Is that test going to be a nasal swab? Is that test going to be in the urine? And once the appropriate test has been decided upon and you've given that test, there's a lot of what we call pre-analytical, steps that happen. Was the right tube chosen? Was the right specimen chosen for the right, specific type of testing, etc. And ultimately, it goes through this toolbox, right? The toolbox Dr. Dean Morado was talking about, where in the laboratories, we have a way to triage our specimens. The labs have microbiologic testing, blood bank testing, hematologic testing, right? So the laboratory could be a simple CBC that you order, or it could be an analysis of the leukemia that might be in a cancer patient's blood. So depending on what we think is the correct approach to testing for the specimen and trying to provide the answer that the physician is looking for, there could be a variety of tests, right? So we have genetic-based tests. We have tests that look at different levels in a particular fluid. We can look for different proteins in a fluid, etc. And then after the machine has sorted it all out, right? We have looked at the machine, it's working fine. And in this mechanism where there's a human interaction and also a machine-based process, there is a whole health informatics process where that data then goes back into your electronic medical record to make its way back to your physician, right? And so this means that at a series of levels, there is a lot of caution that is always taken, right? And so I do not mean to alarm anybody, but, wrong tubes can be chosen. The wrong amount of blood could be given. The nasal swab might not have gone into the place it needed to go to for the test to work, right? There can be analytical errors. There can be specification errors in the machine. There can be errors in the electronic medical record. So the reason I call this a circle of trust is because this works. We have checks and balances on every single step, right? Where what our patients don't understand or see, and they don't have to worry about, is because we take care of this. But what happens to all of this when it comes to a new test, right? It's something that we've never developed before, something that is now a pandemic. Everyone is worried about it, and we have to start developing a test for it. And, since specifically I wanted to talk about COVID-19, if you're thinking of microbiologic tests, right? Testing for bugs, there are intrinsic differences in the way we test for bacteria, for example. I'm showing you a bacterial kind of plate where the bacteria are actually growing on an agar plate. And you can culture bacteria. But on the right, the concept of viral culture is actually very, very difficult. So bacteria are larger organisms, and they can grow on their own. They can make little colonies. They're easier to work with. But viruses are not that easy. Viruses always need another medium that's living in order to propagate. So in order to even culture viruses, for example, if you were to culture them, you would have to inject them into another living medium. And here they're being injected into, let's say, a chick embryo or an egg, right? And so they require that substance around them in order for them to be cultured, right? And that's just one mechanism for viruses to actually be propagated. And within the viral world, there are DNA types of viruses and RNA types of viruses, right? And so I could keep going on, and there's really no need to go deeper than this. However, what is important for us to understand is that the COVID-19 virus on the right is actually an RNA virus, right? And so we start there. Now, let's say that we are a laboratory system, and we actually don't have a way to test for COVID-19, and the pandemic hits, right? And so all of our laboratories are certified by national authorities, and this certification is required for human testing to occur. So we got really well, when this started and the CDC started allowing, the FDA started allowing emergency utilization, allowances. It was very sweet and kind from our colleagues, even our physician colleagues, that were sending us emails saying, I have a PCR machine in my research lab, and we could just start testing there. And I think that really opened up this black box of this concept that even other physicians might not always understand what goes into a test actually being performed, because they've never had to worry about it since the lab takes care of it. But the bottom line is that there is a lot of regulation. In order for that circle of trust to be maintained, there is a significant amount of regulation, and testing for human specimens for clinical results actually has to happen in a very specific type of setting. And there is a lot that goes into it. The specifications of putting a test together require documentation of how accurate this test is, how precise the test is, what the sensitivity and specificity are. And you even need things like positive controls, right? You have to control, how do you know if something is positive? You have to compare it to another positive. And initially, all I can tell you is that, here, our director of microbiology, Dr. Amanda Harrington, she has led this amazing kind of war against, or for testing for COVID-19. And so she's our leader here. But initially, people around the country were looking for positive specimens. They needed a certified positive specimen as a gold standard to even understand whether what they're testing for is actually resulting into a positive test or not. So the way testing is happening right now is under emergency use authorization. And I have a quotation here, and it says that the FDA has not reviewed the test, and that the results from antibody testing, for example, that's one type of testing, should not be used as the sole basis to diagnose or exclude SARS-CoV-2 infection or to inform infection status, right? And so this is just a way that we have been allowed to start doing testing, even though the FDA technically has not cleared it just yet, right? And so most of the testing that is being done right now in hospital laboratories is being done because of this EUA order. So when you think of SARS-CoV-2, I'm going to end with a few slides here. I want everyone in this audience to think about, how would you test for it? Here's the virus, right? And let's say it's in your blood. And let's say it's in the nasopharynx. So what is it do you think would be the best way to test for it, right? You could try and detect some of these proteins, right? Some of these things that they're jutting out from the surface. If you detect them, most likely you have the virus in your system. The other way to do it is to detect the genetic material that is inside the virus, right? The point is that what we're trying to do is detect the virus in your system. So one way to do it is to be able to do it by looking at viral particles, right? These could be proteins that the virus has. It could be the genetic material that the virus has. But you have to understand there's a caveat. So this is the family tree of coronaviruses. There's a lot of coronaviruses, and SARS-CoV-2 is just one of them. So what we have to do is we have to pick out a protein that is exactly the same in only SARS-CoV-2 and not in its other family members. And I guess the example I could give is that let's say you have a large family and we're trying to detect you and not your cousin. And we go by looking at the width of your forehead, right? And turns out that you and cousin Joe both have the same forehead width. Then it's not going to work because we're going to detect the wrong thing. However, if we go for your fingerprint, it'll be different from Joe. So even when we're trying to detect proteins within viruses, we have to be very specific in what type of proteins we're looking for. And so one way to do it is to look for the proteins. And the other way to do it is to actually test the human that is infected for their host response. Every time you are infected with the virus, you will have an immune response to it. And there are ways to detect the immune response. And that is through serologic testing, which Dr. Taylor will be talking about today. So I quickly want to just tell you what it entails in order to be able to pick up a little piece of genetic material from this virus. So first, what we do is we convert the RNA to DNA. And after that, there's an amplification process. This amplification process happens within a machine. And what happens is that a tiny amount of viral RNA is amplified thousands of times and becomes a larger amount, right? So you have to understand this. That means that whatever you swabbed had the virus in it because the RNA was there, and you amplified it to be able to be detected, right? So I want you to draw your attention to the fact that, we've been looking at things like how long does the coronavirus stay on surfaces? It can stay up to 3 hours in airborne state. It can stay up to 72 hours on a glass surface. But what's important to understand is that if you're using this amplification technique, what you're doing is that you're detecting a little piece of a protein and amplifying it. Does that mean that that's actually an infection, or does that mean that it's just the protein hanging around? And this brings us to the question of this genetic testing in patients who are positive weeks and weeks afterwards, right? So this is my friend, Jack Durbin. He and I are Twitter friends. And I've been following his story on Twitter in that, now he's on swab number 8. So this is the nasopharyngeal swab. It goes in, comes out, and then he's still positive 8 weeks later. And the question then is that, does that mean he's still infected? And does that mean he's still infective? And there is recent data to suggest now that just because you detect by amplification procedures this little amount of viral RNA, the RNA is shedding even after the patient has recovered for a little while. And for those of you who are interested in this particular paper that was published very, very recently in Nature, they started looking at days after onset of symptoms. So here's day 0. And what they found was that the cultures from the virus, so let's say they pull out a little piece of viral tissue and they try and see if they can grow the virus from it. After 8 days, right here, there are no more positive cultures, meaning that 8 days after the symptoms started, you can still detect the virus, but that virus cannot become another virus, meaning that hopefully what this early data is indicating is that the infectivity is over. But remember, this is only looking at viral proteins. It isn't necessarily looking at the host response, which will be the second point of our journey. So I'm not going to take any more time. These are a bunch of the machines that we use in our laboratories at Loyola University Health System. Full kudos to our director of microbiology and everyone who's been working tirelessly to make this laboratory test happen and to give accurate data and results to our patients. And just for the segue, I want to show you that what I was talking about in testing is this early viral detection. So after the virus incubates in your body, there's an early acute phase. a late acute phase, and then a recovery phase. And the latter portion of this is actually going to be tested by looking at the host immune response that is basically looking at antibodies. And Dr. Taylor is going to be taking that on. Thank you.
Thank you so much, Dr. Mirza. And I want to just say how fortunate I feel to work alongside you and Dean Murado. I tell people regularly that I feel outmatched by the brilliance of my colleagues here at Loyola, but never so much as listening to your presentation and understanding in greater depth the testing mechanisms. And we're now going to turn to someone who is leading these efforts in public health on behalf of a state that has been at the center of this pandemic. Dr. Taylor, your presentation is here. We look forward to your remarks.
Thank you very much. And I'm very grateful to Loyola University for the opportunity to talk to you. I always learn so much more than I give at these presentations. So thank you very much. So I'm going to talk to you about public health's role in reopening. And you can hear an enormous, I think it's incredibly fascinating that testing has become such a word that is used all the time in our national news. I would never have imagined that this could possibly happen. And so I'm going to talk to you about why it's important and where it should be used and how it should be used. So before I go into my slides, I want to talk to you about the metrics that are going to guide reopening. And Dr. Dean Murado talked about these a little bit in Chicago and Illinois' plan. Essentially, there are 4 things that we have to monitor. There's a thing called R0, or it's sometimes called RT, which is the transmission rate. It's a measure of, if I'm infected, how many people am I going to infect? And what we want to do going forward is to keep that RT or R0 less than 1. We want to make sure that if an outbreak happens, that we have the hospital capacity to admit a resurgence of new cases. And in New York, we want to have 30% of our hospital beds available. We want diagnostic testing to be capable to detect and isolate new cases, so that we want to have a testing capability of at least 30 tests per 1,000 people. And we want to have robust contact tracing. And that's really the theme of what I'm going to talk about, is the fact that these very basic public health principles need to be adhered to guide reopening. Can I have the next slide, please? So as Dr. Misra said, there are 2 types of testing. The diagnostic testing, also called PCR or molecular testing, detects the presence of the viral RNA, and it says you are infected, or if you don't detect the RNA, you are not infected. So that's one type of test. And the other is serology, which detects the presence of antibodies to the virus and confirms that a person was previously exposed. And again, serological testing has been in the press so much lately, and I think there's a great deal of confusion, misunderstanding as to what a positive serology test actually means. So I'm going to talk about the role of these 2 tests in public health and in the reopening policy. Could I have the next slide, please? So why should we do diagnostic testing or molecular testing as we reopen? So if you look in the top left corner, you'll see a guy with a magnifying glass, which is the symbol for doing a test. So if I am tested as positive, I should be told to isolate at home because I'm an infected person and I don't want to spread the virus to anyone else. It's also important that everyone that I have come into contact with is told that I am infectious, and therefore they need to be told to do social distancing, to wear a mask, to watch for symptoms, to do temperature monitoring, and they should be in quarantine for 14 days because that's the period of potential infectivity. The idea is that we want to box the virus in. We want to keep that RT or R0 down to less than 1 so that the possibility of reinfection, of transmitting infection is very, very low. Could I have the next slide, please? This is the detect, trace, and isolate policy. I'm just giving two examples here. There's different policies for how this is done in every country. The one on the left shows an app that is used in Australia at the moment. It's called COVID Safe. And it's an ability to electronically track contacts of people who are infected. It's totally voluntary, but it does speed up the process. And on the right is a system that New York is using a system devised at the Johns Hopkins University in Baltimore, where many, many people are being trained to be contact tracers. So it's a more manual process using an army of tracers, but there are clearly many methods of tracing, and some of them use electronic methods to make the process a little more efficient. But tracing and isolating is incredibly important. Next slide, please. So just Dr. Misra talked about molecular testing, diagnostic testing. Just as a reminder, it's the first tests that were actually developed for the SARS coronavirus. It's based on testing, looking for a sequence of the virus genome. In most cases, it's the nuclear capsid gene, though there are tests available now that detect other parts of the genome. There are many, many tests now, I think over 50 or 60 that are available and are FDA authorized to use. They may be these testing methods may be manual, they may be semi-automated, and the high and low throughput. As I said, there are many FDA approved or authorized tests, and they vary in their throughput. Some hundreds and hundreds a day, others 50, say, a day, and it really depends on the lab that's using them. Can I have the next slide, please? So serological testing. Why are we doing it in New York, and what role does that play in the opening up? Well, I've put 4 bullets of why we're using it in New York. The first one really isn't about opening up, it's about the development of a potential therapeutic. And in history now, there are many examples of plasma donations from people who have survived an infectious disease, where their plasma is actually given to people who are critically ill, and the antibodies in the plasma of people who've survived neutralizes the virus and it's a potential therapeutic to enhance recovery. So that is one reason we're doing it in New York State, to qualify potential plasma donors, to make sure that they have both high levels of antibody and neutralizing antibody. We want to make sure that our first responders are safe. Now, I want to be very careful here. Just because somebody has antibody doesn't mean they're immune. We do not know that yet until we see people being reinfected by the virus and know whether they suffer from disease or whether they do not have disease, is really the only way we're going to know that antibody means immunity. So it's really important to know that. But it's clear that if you don't have antibody, you're not immune. So we want in our first responders to know whether they are developing immunity or not. And I'll show you some data on that in a little bit. we want to monitor exposure in the population. You've heard about the term called herd immunity, so that if there's a high level of antibody in the population, transmission of the virus will be very much slowed. And this is something that ultimately we want to aim at, hopefully with a vaccine, not by everybody getting infected. The other reason we're doing it is that Wadsworth performs basic research, and we want to understand the immune response to the virus. And so we're doing some serology work to understand the immune response. Could I have the next slide, please? Now, how good are the tests? This is something that Dr. Mirza touched on. And as he said, there is no perfect test. Every test has things that are good about it and things that are not perfect about it. I'm not going to say bad, but let's say not perfect. Sensitivity is the, if I say I can detect, if there are 100 things, 100 virus particles in a sample, can I detect 100? And so the closer to 100%, the better. Now, clearly in a disease that's so transmissible like COVID-19, the sensitivity is really important. A false negative, so that somebody is tested and they're really positive, but the test gives you a negative, means that that person is walking in the community and potentially transmitting the virus. So false negatives are really important. Specificity, the impact of cross-reactivity, and I'll show you some data on this in a little bit. There are at least 4 coronaviruses that circulate in the community every respiratory season. These cause the common cold. But there is cross-reactivity between those viruses, the proteins of those viruses, and the SARS-CoV-2. So you want to know if you're detecting antibody, is it to the SARS-CoV-2 or is it to one of those coronaviruses? And then there are basic things like reproducibility. If I get the test twice, will I get the same result? Timing of special specimen collection is really important. There's some evidence now that people are infectious in the period immediately before symptoms occur, and then for a few days afterwards. But if you take the nasopharyngeal swab or a nasal swab too early, person could be positive, but the level of detection of the test is such that you can't detect the virus yet. Or if you take it too late, after the virus is gone, then you may have it be negative even though you were infected. So timing of taking a specimen is very important. And that's also very important for the development of antibody, because you have to take the blood sample at the right time. I'm going to talk about a little bit about rapid tests in a little bit, especially as it relates to serological tests. Could I have the next slide, please? So this is just going back to Immunology 101, and you look at the graph on the left and you can see that IgM is the first antibodies class that appears, and then you get an IgG response, which over time will fall. But then when you get a second exposure to the antigen, the memory response allows the IgG to go up very high and to last a lot longer. And that's the state that we really want people to be in. So there are some serological tests that detect total antibody. So that's IgM, IgG, and another subclass called IgA. And there are some that are IgG specific. And the ones that are IgG specific are probably more useful because they're detecting the longer lasting class of antibody. Could I have the next slide, please? So I talked a little bit about rapid tests. And in serological tests, the cartridge shown at the top of this screen is a rapid test. And you can see that you would load the serum or blood into that little well, and it's called the lateral flow device. And you'll get, first of all, a positive band, which is the first band that you see. And then this test detects IgG and IgM. The problem with these tests is that they don't tell you anything about specificity, nor do they tell you anything about sensitivity. So if this is undiluted serum, you could have a very weak antibody response, which was really not going to be protective, or it could be cross-reactive with other coronaviruses. So the more useful tests really are the ELISA tests, and there are other technologies, but the plate on the right, the 96-well plate on the right, shows you serial dilutions that are used for an ELISA test. And this gives you a much more quantitative approach to serology to be able to understand the level of immune response that you have. So at the moment, there are no antibody tests, rapid tests that are approved by the FDA, only laboratory-based tests. So it's not, there are some of these rapid tests which are, to be perfectly frank, very cross-reactive and not good, but there are no doubt are some good tests, but in this situation, they are of limited value. Could I have the next slide, please? Now, here's as techie as I'm going to get. This is a study that we have done in our lab using, we don't use ELISA, we use something called a Luminex-based microsphere assay, but that doesn't matter. On the red dots on the left are sera from patients who had a known PCR positive to COVID-19. So we know these are positive patients. And the blue dots on the right are sera from somebody who was positive to the coronavirus NL63, to influenza, to metapneumovirus, and to other respiratory viruses. And you can see that in this assay, there's a great deal of cross-reactivity at quite low levels to the SARS-CoV-2 antigen that's used in the test. And so we have set what's called our baseline at 6 standard deviations above the baseline so that you eliminate all that cross-reactivity. But by doing that, we also lose some sensitivity. So all the little red dots on the lower right are people who are actually positive to COVID-19, but we're not detecting them because they're in the range of cross-reactivity. And all the dots upward in the graph are people who were collected later in an infection where their antibody level has risen. So specificity and sensitivity are really important to know about in a test because they make interpretation it makes the interpretation very important. We know we're losing some sensitivity, but it's important for us to just be very aware of the cross-reactivity. Can I have the next slide, please? So when you're interpreting serological assays, we know that they're good for seroprevalence studies, and I'll show you some data on that in a minute. And they are important for saying, yes, I was infected previously. But we do not know enough yet to say I'm immune and I can go back to work without using protective equipment or wearing a mask or maintaining distance. So certainly more study is needed in this area yet in interpreting serological assays. Can I have the next slide, please? So we have an assay that we developed at the Wadsworth Center where we actually use blood spots collected onto filter paper as our source of blood. And we're using them to do seroprevalence studies in the community. And these are some data. We've actually done well over 40,000 of these now in different populations. And a random population shows that our prevalence is anywhere between 14% and nearly 20%, and that 20% level is in the New York City metro area, whereas the 13% is upstate. In our first responder populations of the fire department and the police department, we're between 10 and 17%, again, downstate. Our essential state workforce, including transit workers, around 14%. And in healthcare workers, which of course is an incredibly important and sensitive population, around 12%. Now, these studies have been done in the last few weeks, and clearly it's going to be important to repeat these studies multiple times as we go forward, because what we are going to be looking for is the seroprevalence increasing? And they're going to be important also when a vaccine is made, to see the response in individuals. But this gives you a sense of which populations may be most vulnerable. Could I have the next slide, please? Oh, that's a repeat. Could I have the next slide, please? So now, just as I finish up, I just want to talk about reopening. This is a plan. We're beginning reopening in New York now, and the governor has put out a template for business reopening. And each business that reopens has to have a plan that addresses people, places, and processes. So for instance, what is your policy going to be about physical distancing? Are you going to make sure that all people have temperature monitoring when they enter the workplace? What are you going to do if somebody has an elevated temperature over 100? How are you going to institute physical distancing in meeting rooms and things like that? So each business is going to be required to have a plan. And if you're looking for a template to adapt, this is actually quite good. It's very straightforward. And then one last thing in the next slide. This is, for me, recommended reading. Atul Gawande from Brigham and Women's is one of my public health heroes. And he recently wrote this article in the New Yorker, which to me should be required reading. It talks about how his hospital, Brigham and Women's, has worked during the COVID outbreak, how they have developed processes for inquiring about symptoms, about temperature checking, about social distancing, about hand hygiene, the importance of washing hands. And the other thing that he talks about is the culture of the workplace, how we're all a bit reticent to say to somebody, I need to stand distant from you, or you're not wearing your mask, or could you put your mask on? And we're now going to be in a culture going forward where it's going to have to be okay to say, please wear a mask if you talk to me, or please wash your hands more frequently. And so he really brings up the importance of not only population responsibility, but personal responsibility that we all have to take for not only our own safety and wellness, but the wellness in the community of people that we work with and people we love. And so it's a different way of thinking of personal responsibility. I think that something we're not totally used to, that can be a bit intrusive, but something that we have to get used to. Next slide, please. And that's the end of my presentation, and I'm happy to have any questions. Thank you.
Thank you so much, Dr. Taylor, and thank you to all of the presentations. I'm actually going to stop sharing the slides now so we can see each of you in a little bit more of a close form. And Dean Morano, let me start the questions with you. One of the things that we've seen in the chat box is a lot of people wondering about the last points that Dr. Taylor was talking about. What should workplaces be thinking about in their companies or organizations? One question we got in the chat box, for example, should workplaces themselves be thinking about organizing tests? So just high level to get to Dr. Taylor's idea of how do we keep that R below 1, where we're really lowering that transmission. For the many people out there who are involved in their workplace operations, what tips do you have? And then we'll go to the other panelists.
Yes, I'd like to draw people's attention back to I think it was the second to last slide that Dr. Taylor mentioned, which was a template that is coming from New York State. I think those types of templates are a systematic way of walking through the questions of how do you take the practice and adapt it for your own local setting in the questions. And I believe rather than every company trying to figure out how do I test, it's more or less how do we plug our employees into the systems that are around us, and the scaling up of testing that's in community. I think most small businesses are not going to be expected to have that capacity to be doing local testing, but they can be promoting the culture and having processes in place that create a culture of wearing masks, of protecting people if they need to be taking time off or not, and isolating in that. So that would be my advice, to look at that template and use that as a guide to build your own individual plan. When I talk to my brother who runs a small business, that's essentially how I walked him through that. And every employer is going to have a different scale or a different setting, and it really needs to be tailored to your own local work environment.
Dr. Mirza, if I can go to you next, one of the other sets of questions we've received in the chat is around just the timing of testing when you're thinking about, when you test, when the result comes back, when you're notified. I know that varies, but if you can maybe give us guidance, especially on what you're seeing in Illinois.
Sure. So I think I can speak towards our university health system, which, is just one model of doing it. And so naturally at least in our health system, there is a triage line. There's a certain amount of pre-test positivity probability, in the sense that, if you have symptoms, let's say you're working in a healthcare environment, we're all being screened for fevers when we walk in. And the testing is being done in a very kind of controlled fashion based on what type of symptomatology that you have, and, what type of floors in the hospital that you're dealing with. And I think to directly answer that question, testing specific, like the testing methodology does not necessarily take that long. There are most places, if you're not talking about point of care tests, which I know somebody also mentioned in the chat, but the usual PCR, et cetera, should be reported very soon. So I know that somebody had mentioned there was a 9-day delay in letting a patient know. I think that was probably one unfortunate kind of one-off situation. But most tests are done and reported within 24 hours. There is a point of care test, which is, a test in which you can probably get a result in 15 minutes. There are some hospital systems using that too, but all these tests are being used in very specific situations. For example, let's say elective surgeries have restarted and we need to now test asymptomatic patients, for example, right? And so there are different tests for different types of situations, and the resulting timeframes are a little bit variable, but usually what has happened is that if people are tested, then they're supposed to be in quarantine until they get the results back.
Dr. Taylor, if I can come to you, quite a few questions in the chat were around public health guidance. There are a number of parents in the webinar who are wondering about their kids playing with other kids, and if there, is guidance over time on how to think about those issues from both, I guess, a public health, but also a mental health standpoint.
I think that we have to make risk-based decisions. So if you're going to have, I wouldn't have a whole bunch of little kids invited to my home and playing in the living room. So take them for a walk in the park and let them run, and, you know, make a game out of staying 6 feet apart. You know, this world that we have now, it's changed us. I think a level of fear and uncertainty that I've certainly not experienced in my life so far. And, you know, I think of young people. So we're going to have to make up new rules for how we go forward for at least the next period until we get a vaccine. And so, you know, use common sense, use public health guidance, wash your hands all the time, be careful, you know, be aware of surfaces that you should disinfect. It's fairly basic stuff that we can really use to make a difference in the infection rate, but we've got to be sensible. And we've also got to understand that, you know, we're sensitive beings who need to be happy as well as isolated. Yeah.
So we have about 3 minutes. I want to have a power round ending. So 60 seconds each. Tell us about what kind of leadership people need to display in this moment to best help their companies and their culture. It's something that's been brought up throughout, and I'm just curious for each of you to share what you think that leadership looks like, since we have so many executives in the webinar. And maybe, Dr. Shell, we'll come back to you, and then we'll go in backward order this time, and then with Dean Murado. So Dean, Dr. Taylor, you're first.
Oh, yep. Common sense, public health, consistency, data-driven decisions, not emotional decisions. A leader displaying that.
Thank you. Dr. Mirza?
I fully second that. Data-driven decisions, and to me, I think very open, like open communication, right? I mean, everybody understands that we're all learning. We're all learning at the same time. This is a completely new experience for everyone at every level. And so data-driven decisions and open communication, whatever the situation is, communicate.
Dean Murado, as a leader yourself?
Yes, and I'll do a segue to next week's one, which is how do we use these data and the evidence to create adaptive systems? You know, you might say a beginner's mindset. If you had to start and take this knowledge with a beginner's mindset, the entrepreneur, how do we now be flexible and create new systems? And then the other would be, you know, search and reapply. How do we learn from other systems like New York and others that have been trying it? And just good marketing skills is seeing what competition is doing. Well, there's lots of public health competition going on, and we can search and reapply from best use cases to ours. And so I also saw a lot of comments, Seth, around people looking for those kinds of resources. And maybe that's something you and I can do in advance of next week's meeting, is to pull some of those together so folks don't have to do all of the Google searching on their own. We can guide them to some credible sources.
Well, I want to applaud our panelists and just say how, I mean, truly insightful all three of you were in giving us both actionable tools and true background on the health processes. And I just want to end by saying thanks to you and to everyone on this webinar. A lot of you are on the front lines, and you are truly allowing us to be resilient in this pandemic. And so thank you for what you're already doing, and let's hope that we together can continue to keep that R much less than 1 until we hopefully get to a vaccine that gives us the right type of herd immunity, as we talked about earlier. Thanks to everyone for being part of this, and hopefully we'll see you next week when we continue the discussion. Thank you.
Thank you.